गुड आफ्टरनून स्टूडेंट्स। उस दिन आपको पढ़ा रहे थे लेटेस्ट ट्रेंड्स इन ऑटोमेशन, उसी को कैरी फॉरवर्ड करते हैं। आज आपको पहले बताते हैं माइक्रो एरे के बारे में। जो बायोचिप्स लेटेस्ट मशीन्स में यूज होती हैं उसमें जो सिस्टम यूज होता है वो है माइक्रो एरेज। द dense two-dimensional grid of biosensors is a critical component of biochips. Typically, the sensors are deposited on a flat surface which may either be passive like silicon or glass or active consisting of integrated electronics or micromechanical devices that perform or assist signal transduction. Microarrays are not limited to DNA analysis. Protein microarray, antibody microarray, chemical compound microarray can also be produced using biochips. Randox Laboratories Limited launched their first protein biochip array technology as Evidence. मशीन का नाम था Evidence. Randox Laboratories ने इसको लॉन्च किया था in the year 2003. In protein biochip microarray technology, the biochip replaces the ELISA plate or the cuvettes as the reaction platform. The biochip is used to simultaneously analyze a panel of related tests in a single sample, producing a patient profile. The patient profile can be used in disease screening, in the diagnosis, prognosis, monitoring the disease progression, or monitoring the treatment. Biochip array technology is a novel application of a familiar methodology using the sandwich, competitive, and the antibody capture immunoassay. In sandwich assay, an enzyme-labeled antibody is used, while in an competitive assay, an enzyme-labeled antibody is used, and on the antigen binding chemiluminescence reaction produces light. जो एंटीजन एंटीबॉडी रिएक्शन के कारण लाइट उत्पन्न होती है उससे हम अपने एनालाइट की कंसंट्रेशन को हम नाप सकते हैं। डिटेक्शन इज बाय अ चार्ज्ड कपल डिवाइस और अ सीसीडी कैमरा। द सीसीडी कैमरा इज अ सेंसिटिव एंड हाई रेजोल्यूशन कैमरा with sensors able to accurately detect and quantify low levels of light. The test regions are located using a grid pattern. Then the chemiluminescence signals are analyzed by the imaging software to rapidly and simultaneously quantify the individual analytes. अब हम बात करेंगे अगली रिसेंट एडवांसेस इन ऑटोमेशन की, जो होते हैं नैनो सेंसर्स। बहुत सी नई मशीनों में भी ये इनकॉर्पोरेटेड होते हैं नैनो सेंसर्स। नैनो सेंसर्स आर एनी बायोलॉजिकल, केमिकल और सर्जरी सेंसरी पॉइंट्स यूज्ड टू कन्वे इंफॉर्मेशन अबाउट नैनो पार्टिकल्स टू द मैक्रोस्कोपिक वर्ल्ड। दो humans have not yet been able to synthesize the nanosensors. Predictions for the use mainly includes various medicinal purposes and as gateways to building other nanoproducts such as computer chips that work as nano robots. The medicinal uses of nanosensors mainly revolves around the potential of nanosensor to accurately identify particular cells as places in the body in need. By measuring changes in the volume, concentration, displacement, velocity, gravitational or electrical or magnetic forces, the pressure or the temperature of the cells in the body. Nanosensors may be able to distinguish and recognize certain cells, most notably those of the cancer at the molecular level, in order to deliver medicine or to monitor development to specific places in the body. One example of nanosensor involves the fluorescence technique of the quantum cadmium selenide dots to uncover the tumors within the body. By injecting a body with these quantum dots, a doctor or a physician could see where a tumor or cell was by finding the injected quantum dots, which is an easy process because of its fluorescence. जहां कहीं भी कैंसर सेल्स पाए जाएंगे, जो कैडमियम सेलेनाइड के क्वांटम डॉट्स होंगे, वो वहां पे रेडियो इमेजिंग के द्वारा फ्लोरेसेंस उत्पन्न करेंगे और रेडियो इमेजिंग टेक्निक से हम उसको वहां पे कैचअप कर सकते हैं किस पर्टिकुलर ऑर्गन में या किस पर्टिकुलर सेल रीजन में वहां पर कैंसर फैला हुआ है. Developmental nanosensor quantum dots would be specifically constructed dots to find particular for which the body was at risk. A downside to the cadmium selenide dots is that they are highly toxic to the body. As a result, researchers are working in developing alternate dots made out of a different, less toxic material while still retaining the fluorescence properties. There are several proposals to make nanosensors including top-down lithography bottom assembly and molecular cell assembly. Certain electromagnetic sensors are being used in photoelectromagnetic systems also nowadays. The development and miniaturization of these nano biosensors should provide interesting new opportunities. हाँ, अब ये जो नैनो सेंसर टेक्नोलॉजी है, ये नई जनरेशन के लिए नई हो सकती है और थोड़ी सी कॉस्टली भी हो सकती है, लेकिन इसके रिजल्ट बहुत ही प्रिसाइज और एक्यूरेट आते हैं. इसलिए पैथोलॉजी लैब में कई सारी मशीन्स में ये टेक्नोलॉजी इनकॉर्पोरेट होके ऑटोनाइजर सिस्टम्स में आजकल यूज की जा रही है. नैनो सेंसर टेक्नोलॉजी इज अ रिलेटिवली न्यू फील्ड एंड द ग्लोबल प्रोजेक्शंस फॉर साइट्स ऑफ प्रोजेक्ट्स incorporating nanosensors ranges from $6 billion to $12 billion in the next 3 to 4 years. Nanosensor developers must overcome the present high cost of production in order to become worthwhile for implementation in the consumer products. Nanosensor reliability is not yet suitable for the widespread use. Nanosensors have yet to be made compatible with most consumer technologies for which they have been projected. जो नैनो सेंसर टेक्नोलॉजी है, एक नई प्रकार की तकनीक है, नव विकसित टेक्नोलॉजी है, जो धीरे धीरे से हमारी नई जनरेशन को पता चल रही है और कई सारी जो पैथोलॉजी लैब के इंस्ट्रूमेंट्स हैं, ऑटोनाइजर्स हैं, उसमें इसको यूज किया जा रहा है. हाँ, थोड़ा सा ये कॉस्टली अभी पड़ रहा है, तो इस पर रिसर्च चल रही है कि इन मशीनों के दाम जो नैनो सेंसर टेक्नोलॉजी पर बेस्ड है, उसके दाम को कैसे घटाया जाए. नेक्स्ट लेटेस्ट ट्रेंड ऑफ ऑटोमेशन में आता है लैब ऑन अ चिप. अ लैब ऑन अ चिप और एन एलओसी इज अ डिवाइस दैट इंटीग्रेट्स वन और सेवरल laboratory functions on a single chip, only millimeters to few square centimeters in size. The lab on a chip technology deals with the handling of extremely small fluid volumes down to less than picoliters. The lab on a chip devices are often indicated by micron total analysis system or MUTAS system. The lab on a chip is general scaling of a single or multiple laboratory processes down to a chip format, and the micro total analysis system is dedicated to the integration of the total sequence of the lab processes to perform chemical analysis. The microarray technology was started in the year 1954 for realizing the integrated semiconductor structure for developing the electronic chip. This technology soon was applied on pressure sensor manufacturing. Due to further development of these small compatible processes, a toolbox became available to create micrometers in silicon wafers as well. The micro electromechanical system with the microsystem technology as an LOC analysis system was firstly used as a gas chromatograph, developed in 1975 by Scattery of the Stanford University. However, only at the end of the 1980s and the beginning of the 1990s, the LOC research started to have a serious growth. A big boost in the research and commercial interest came in the mid-1990s when this new task technology turned out to be providing interesting tools for genomic applications like capillary electrophoresis and DNA microarrays. A big boost in the research support also came from the military and the defense advanced organizations, research projects agencies for their interest in portable biochemical warfare agent detection systems. Some advantages of LOC are that these LOCs may provide less or low fluid volume consumption, that is less waste, low reagent cost and less required sample volume for diagnostics. LOC मशीनों पर जो हमारा रिएजेंट का वॉल्यूम होगा और जो हमारा सीरम का रिक्वायरमेंट होगा वो अपेक्षाकृत जो मैनुअल मेथड है उससे बहुत ही कम मात्रा में रिएजेंट और हमारा सीरम यूज अप होगा, इसलिए ये टेक्नोलॉजी हमारे लिए फायदेमंद रहती है. नेक्स्ट पॉइंट, फास्टर एनालिसिस एंड रिस्पॉन्सेस टाइम ड्यू टू शॉर्ट डिफ्यूजन डिस्टेंसेस, फास्ट हीटिंग, फास्ट सरफेस टू वॉल्यूम रेशियोज, better process control because of a faster response of the system like thermal control, compactness of the system due to integration of much functionality and small volume. छोटी सी मशीन होती है, इसको हैंडल करना ज्यादा डिफिकल्ट नहीं होता है अपेक्षाकृत बड़े बड़े ऑटो एनालाइजर सिस्टम से. Lower fabrication costs allowing cost effective disposable chips, and it is a safer platform for chemical, radioactive or biological studies because of integration of functionality and smaller fluid volume. ये तो थी लैब ऑन अ चिप की अच्छाइयां, एडवांटेजेस. अब आते हैं इसकी डिसएडवांटेजेस पर. Disadvantages of LOC are that novel technology and therefore not yet fully developed. The physical and chemical effects that become more dominant on a small scale sometimes make the process in the lab on a chip behave more complex than in the conventional laboratory equipments. कभी कभी जो इनकी हैंडलिंग होती है वो हमको पुरानी जो लैब मशीनें यूज होती हैं उसके अपेक्षाकृत हमको थोड़ा सा नयापन लगता है और उसको समझने की हमें कोशिश करनी चाहिए. इंजीनियर के द्वारा या इंटरनेट के द्वारा हमें सीखने की जरूरत पड़ती है इस मशीन को कैसे आगे ऑपरेट करना है. इसलिए एक नॉर्मल लैब टेक्नीशियन के लिए कभी कभी से ज्यादा उपयोगी नहीं रहती है. द डिटेक्शन प्रिंसिपल्स मे नॉट ऑलवेज स्केल डाउन इन अ पॉजिटिव वे. कभी कभी फॉल्स नेगेटिव, फॉल्स पॉजिटिव रिजल्ट्स भी आने लगते हैं LOC पर और इसलिए कभी कभी ये पूरी तरह से भरोसेमंद टेस्ट हमारे लिए नहीं रह जाता है एक पैथोलॉजी लैब में. Although the absolute geometric accuracies and precision in the microfabrication is high, they are often poor in the relative ways compared to precision engineering. अब हम अगली टेक्निक की बात करते हैं जो लेटेस्ट ट्रेंड्स में आता है, that is immunonephelometry. This quantitative technique has replaced radial immunodiffusion, particularly when large number of samples are to be assayed. The principle of the assay lies in the fact that when dilute solutions of the antigen and the antibody are mixed under antibody excess conditions, the soluble antigen antibody complexes are formed that remain in the suspension. The relative amount of soluble aggregates that can be assayed is by measuring nepherometrically the amount of the light dispersed by the mixture. This is most frequently done by passing a beam of light through tubes containing mixtures of fixed amounts of antibody and variable concentrations of antigen. The scattered light is measured at angles varying from near 0 to 45 degrees relative to the incident light. If the antibody concentration is kept constant, the amount of the dispersed light will be directly proportional to the antigen concentration. The principles of calibration and extrapolation of unknown values are identical to those of the radial immunodiffusion. Immunoflowmetry is superior to radial immunodiffusion in terms of sensitivity, speed, and automation. However, the preparation of samples and cost of equipment and reagents makes it mostly suitable for large volume assays. In the right setup, it is the technique of choice for the measurement of immunogenic proteins such as immunoglobulins and the complement components in concentrations greater than 5 to 10 milligrams per liter. Next technique आते हैं हम immunoelectrophoresis. Immunoelectrophoresis is a two-step technique that combines electrophoretic separation and double immunodiffusion. In the first step, a protein mixture such as serum is electrophorized through a support medium, usually agarose. After electrophoresis is completed, specific anti-serum are deposited in a trough cut into agarose parallel to the axis of the electrophoretic separation. The electrophoretically separated proteins and the antisera diffuse towards one another, and at the zone of antigen antibody equivalence, precipitate patterns form in the form of arcs appear in the reaction. This technique was widely used for analytical studies in patients suspected of plasma cell malignancies. It has been largely replaced by immunoblotting methods, the interpretation of which is much simpler. Next method हम आते हैं, that is immunoblotting. Immunoblotting को हम immuno fixation का भी नाम दे सकते हैं. The principle of immuno fixation or immunoblotting is one of the separation lines is stained as a reference for the position of different serum proteins, while paper strips embedded with different antibodies are laid over the remaining separation lanes. The antibodies diffuse into the agar and react with the corresponding immunoglobulins. After washing off the unbound immunoglobulins and the antibodies, the lanes where immunofixation takes place are stained, revealing whether the antisera recognized the proteins they were directed against. The main application of immunoblotting is the detection of monoclonal proteins. Western blotting is a variation of immunoblotting. इसी का एक प्रकार होता है वेस्टर्न ब्लॉट जिसको हम कहते हैं. It is a variation of immunoblotting that has been popularized for its use in the diagnosis of HIV infection. The first step in the preparation of an immunoblot, which is also known as the Western blot, is to separate the different viral antigens according to their molecular size. This is achieved by performing electrophoresis in the presence of negatively charged detergents such as sodium dodecyl sulfate, which becomes associated with the proteins and obliterates their charge differences, using as support for the separation of medium with seeding properties. The result is the separation of a protein mixture into components of different sizes. After the separation of the viral proteins is completed, it is necessary to transfer the separated proteins to another support in order to proceed with the remaining steps. This transfer or blotting is easily achieved by forcing the proteins to migrate into a nitrocellulose membrane by a second electrophoresis step or electroblotting. The nitrocellulose membrane to which the viral antigens have been transferred is then impregnated with patient's serum. If antibodies to any or several of these antigens are contained in the serum, they will combine with the antigen at the point where it is blotted. The following steps are designed to detect the antigen-antibody complexes formed in the cellulose membrane. Firstly, all the non-precipitated antigens and the antibodies are washed off, and then the protein binding sites still available on the membrane are blocked to prevent false positive reactions. Finally, the membrane is overlaid with a labeled antibody to human immunoglobulins. This antibody will react with the complexes formed by viral antigens and human antibodies, and later can be revealed either by adding a color or chemiluminescence reaction developing substrate, that is, the anti-human immunoglobulin is labeled with an enzyme, or by autoradiography. The reaction of the labeled antibody in the sites is corresponding to the separation of the viral proteins, indicating that the patient's serum contains antibodies reacting with those of the antigens. This technique has the advantage over the screening assays of not only detecting antibodies but identifying the antigens which the antibodies were directed. This results in increased specificity. The downside of this is that time consumed during the assay running, which requires very careful quality control and needs to be carried out by specialized personnel in certain pathology laboratories. Western blot membranes are nowadays purchased commercially. These specific blocks or blots for especially HIV identification need only to be proved with the patient's serum and developed. Generally, positive and negative controls are included with the blots. Using these commercially available kits, the performance of sophisticated technology is accomplished with less time and training. नेक्स्ट मेथड अब हम बात करेंगे लेटेस्ट ट्रेंड्स इन ऑटोमेशन में। काउंटर इम्यूनो इलेक्ट्रोफोरेसिस की। काउंटर इम्यूनो इलेक्ट्रोफोरेसिस टेक्निक is a variation of double immunodiffusion, in which antigen and the antibody are forced to move towards each other with an electric current. The antibodies move to the cathode, while most antigens which have a strong negative charge move towards the anode. Precipitin lines can be visualized between the antigen and the antibody wells. This method is faster and more sensitive than double immunodiffusion. Visible precipitation can be usually observed after 1 to 2 hours, although for maximal sensitivity it is necessary to wash, dry and stain the agar gel in which the reaction took place, a process that extends the time to final reading of the results to several days. कई सारे दिन भी ले सकते हैं, इसलिए ये थोड़ा सा लैबों में अब कम यूज होता है। Counter immunoelectrophoresis has been used for the detection of fungal or bacterial antigens in CSF, that is of the patients with suspected meningitis, and for the detection of antibodies to Candida albicans and in DNA and other antigens. Next is radial immunodiffusion. Radial immunodiffusion represents a hallmark in the evolution of immunoserology because it represents the first successful attempt to develop a precise quantitative assay suitable for routine use in the diagnostic laboratories. Radial immunodiffusion receives its designation from the fact that a given antigen is forced to diffuse concentrically on a support medium in which antiserum has been incorporated. पहले से ही एंटीसीरम उसमें कंपनी ने लगाया हुआ होता है जिस भी एंटीबॉडी के में टेस्ट करना होता है. A polyclonal antiserum known to precipitate the antigen is added to molten agar and an agar plate containing the antibody is then prepared. Identical wells are cut in the antiserum containing agar and those wells are filled with identical volumes of samples containing known amounts of the antigen or calibrators and unknown sample in which antigen needs to be assayed. After 24 to 48 hours, it is possible to measure the diameters of the circular precipitates formed around the wells where antigens were placed. Those diameters are directly proportional to the antigen concentration. A plot of the precipitation ring diameters versus concentrations is made for the samples with known antigen concentrations. This plot is also known as the calibration curve and is used to extrapolate the concentrations of antigens in the unknown samples based on the diameter of the corresponding precipitant rings. Radial immunodiffusion, while not as sensitive as some newer technologies, is relatively slow and nonetheless reliable for routine assays of many serum proteins. It has been widely applied in the assay of immunoglobulins, complement components, and in general for any antigenic protein that exists in concentrations greater than 0.5 milligrams per liter in any biological fluid. Next is quantitative immunoelectrophoresis, also known as rocket electrophoresis. Quantitative immunoelectrophoresis is an adaptation of radial immunodiffusion in which the antigen is actively driven through an antibody containing matrix by an electric potential. As the equivalence zone of the antigen-antibody reaction is reached, the reaction ceases and elongated precipitant arcs, पतली पतली सी रेखाएं जो भी एमिनो एसिड की होती है वो उसमें हमको रिएक्शन में दिखेंगी. As elongated precipitant arcs or rockets become visible. The length of these rockets is proportional to the antigen concentration in the wells. Rocket electrophoresis can be used for the quantitative assay of many proteins including immunoglobulins. The reverse modality with antigen incorporated in the agar can be used for the measurement of specific antibodies, and this technique of quantitative immunoelectrophoresis is faster and more sensitive than radial immunodiffusion. अब नेक्स्ट मेथड की हम बात करते हैं जो होता है इलेक्ट्रोफोरेसिस. इलेक्ट्रोफोरेसिस refers to the migration of charged solutes or particles in a liquid medium under the influence of an electric field. In other words, movement of the charged particles because of an external electrical field is called as electrophoresis. Essentially, electrophoresis is a separation technique just as chromatography or ultracentrifugation, and there are similarities among these techniques. In electrophoresis, the electric field is applied to a solution through oppositely charged electrodes placed in the solution. A particular ion then travels through the solution towards the electrode of opposite charge. Thus, positively charged particles or cations move to the negatively charged electrode or the cathode, while the negatively charged particles or anions migrate to the positively charged electrode or the anode. As different charged molecules move, they can be separated if they have different velocities in an electric field. The first electrophoresis method used was the free solution or moving boundary method devised by Tiselius in 1937. The earliest separations using the Tiselius apparatus resolved the serum proteins into 4-component mixtures with the alpha-1 fraction incompletely separated from albumin. This technique is still used in the research for the measurement of electrophoretic mobility and for the study of protein-to-protein interactions. Now coming on to the theory of electrophoresis. In brief, the theory of electrophoresis can be described as the phenomenon of chemical species carrying an electrical charge by virtue of ionization moving either to the cathode or the anode in an electrophoresis system depending on the kind of the charge they carry. The rate of electrophoretic migration is dependent on the factors such as net electrical charge of the molecule, size and shape of the molecule, electrical field strength, properties of the supporting medium, and temperature of the operation. The pH changes the charge of the analyte and its mobility, and it can also affect the structure of the analyte. The ionic strength changes the voltage or the current. Increased strength reduces the migration velocity. Too high current causes overheating. The migration velocity is proportional to the voltage. Overheating can denature the proteins, and reduced temperature reduces diffusion but also the migration velocity. Band resolution increases linearly with the time. The pore size and endoosmosis can affect the migration velocities. Some factors which affect the electrophoretic mobility are the force on the particle. The force exerted on a charged particle depends on the electric field in volts per centimeters, while the charge on the particle is denoted by Q. However, a particle moving in a solvent will experience resistance because of viscosity of the solvent. The resistance, which is itself a force, is proportional to velocity and the proportionality constant F, called as the frictional coefficient. The frictional coefficient depends on the viscosity of the solvent and the size and shape of the particles. The greater the viscosity, the slower is the movement. जितना गाढ़ा सॉल्यूशन होगा उतना उसमें मॉलिक्यूल्स का आपस में मूवमेंट बहुत कम हो जाएगा. The bigger or more asymmetrical the particle, the slower is the movement through the solvent. The frictional coefficient of a large particle such as a protein is characteristic property of the particles. Another factor which affects the electrophoretic mobility is the mobility of the particle. When an electric field is applied to the charged particle, the particle will begin to migrate. The electrophoretic and frictional forces oppose each other, and the particle's velocity increases until the forces are equal. The velocity of a particle attains for a given electric field is determined by two properties of the particle, its charge and its frictional coefficient. Another factor which plays an important role is effect of the pH on the particle. Each ion has a particular charge and mobility. However, when a solution contains a substance which is near the pH of the solution, the substance exists as a charged and as an uncharged form in the solution. Since the effective or average charge of the substance varies with the pH, its effective mobility also varies with the pH. In electrophoresis, a substance such as protein is put in one limited region or zone or of the system and it is made to move into another region or zone. Therefore, much of the solution will not have protein in it at any particular time. If the protein and its associated counter ions are not present to carry the current in a particular region, other ions must be present to carry the current. For this reason, it is a common practice to add an excess of about 0.1 m of low molecular weight buffer to the solution through which the proteins must travel. The buffer and the salt ions or the electrolytes provide a constant electrical environment so that the overall movement of the protein will be as constant as possible and will be minimally influenced by other protein molecules. चलिए बच्चों, आज के लिए इतना ही. नेक्स्ट क्लास में इसको फर्दर पढ़ाएंगे. थैंक यू.